Hey friends, I wrote a paper. I'm really proud of it. It's like I'm a real scientist or something like that. The paper's called Macrofungi from the Hebron and Jerusalem Hills of Palestine. There's me, Teresa, I wrote it, and I wrote it with the assistance of the Palestine Museum of Natural History. I hung out there for like a month and went looking for mushrooms. They're really pretty, see? Here's a bolete, and this one over here, this guy, he's called the scurfy twiglet. Oh, he's so cute. There's a bird's nest fungi here. Yeah, look at that. That's crazy. This thing exists in the world. So I wanted to make a video about how I wrote this paper so that maybe one day you could write something similar. I'm actually going back to the Palestine Museum of Natural History next winter 2021, and you could come with me if you want. I'm teaching a class through Binghamton University, and we're gonna write another paper about Palestinian fungi. It's gonna be great. But before I can tell you about mushrooms, first I have to tell you a little bit about Palestine from the air, for context. So this is the wall. You might have heard of it. Construction began in the early 2000s during the Second Intifada, and it's been ongoing ever since. You can actually see this section curving around the mountain to isolate this valley. They were building more wall there last year when I was gathering mushrooms, and I don't expect I'll be able to visit this valley again, at least not from the Palestinian side. The wall grabs up land like that. These are olive groves that belong to the people who live in these houses on the ridge. These are groves that have been somewhat collectively managed for generations, and it's places like this where I gathered my samples. And here you can see this walled highway reaches over the valley and then tunnels under the mountain. The highway is a settler road. Palestinians aren't allowed to drive on it, and it connects Israeli settlements deeper in the West Bank, like Neve Daniel, here directly to Jerusalem, so that settlers can bypass the older roads that lead through Bethlehem and other Palestinian areas. You can see the outline of the wall here in light brown. So the crazy thing about this bridge is that on the Palestinian side there's a tall wall, you see, so that the drivers on the bridge can't look over to see Palestinian buildings. But on the Israeli side there is no wall, so all the drivers can see is this beautiful Kirmizan valley. And the wall snakes up the hillside over here until the valley is completely enclosed. One thing about the occupation that most Americans don't really understand is how it can be nearly invisible on the Israeli side, yet totally constraining for Palestinians. Like, these olive groves used to belong to the people who lived in these houses before the wall was built. The IDF built that wall so that it wouldn't really be visible to Israelis, but it would also capture as much Palestinian land as possible. And that fact means that this is going to be a pretty partisan video. I guess I should explain here that I'm Jewish, Ashkenazi. My grandparents' entire families were exterminated in Auschwitz. So I understand the allure of this myth of Israel as home. That's why I gather mushrooms there. But one thing that mushrooms have taught me is the value of looking for hidden things, underground things. So in this video, I'm going to show you Palestine as I saw it, from one particular perspective and shot on one side of the wall. So, let's go on a mushroom hunt.
So, we're here in Bethlehem at the Natural History Museum. We're at the Natural History Museum, and we're going basically to Hargila, Wadi Amachor. Great. So we parked the car at the top, and now we're going to go down into this valley here. What do you think this is, this structure here? That's amazing. Alright, so we're looking for mushrooms. It rained yesterday. We're gonna look in this valley here and see if we find anything interesting. First one, just at the base, right there. There's two species of mushroom here. These little guys, they grow on olive leaves. Yeah, you see it grows straight out of the olive leaves. This mushroom comes up just after it rains. What is this one? We will have to look it up. So with this species, there's a clear transformation in the color of the cap as the mushroom matures. So it goes from blackish to brown to a lighter brown.
One of the most used non-cultivated plants in the West Bank.
Arum Palestina, you say?
Yeah.
Can we munch on it?
Yeah.
Yeah?
I don't know if we can eat it raw. We can try.
Okay, let's see. Huh?
What do you think?
Tastes good.
Yeah?
Yeah. That's where I set up the time lapse. Another real mushroom, rare mushroom. What does it say?
White star.
Cartridge, 40mm white star parachute, tear gas. God damn. Cave. It's very warm in here. The world out there. Okay, we found, how many mushroom species have we found?
A lot.
Yeah, like, I don't know, a dozen or so. Now it's time for lunch. Hi. Hey, friend. Don't have that much food for you. Gonna set up a 360 time lapse in this old, probably Ottoman dwelling or shed or something. This is a very old limestone structure. What do you think this structure was? Think someone lived here? Look at this headstone. Cave in the house. What would this have been for? Maybe it was like an agricultural storage shed? What? Who lived here? What happened here? In the threshold. Mushroom in the threshold. Hello. There's a bolete. That's totally an old bolete.
So that was a mushroom hunt. I changed outfits, by the way. I filmed this over several days. Please forgive me. So this is Bethlehem here, and I was mainly gathering mushrooms in these three valleys. This one is Kirmisan, and here's Al-Mahrour, where that vlog was shot, and this one, Husan. And all three of these valleys are located in what's called the seam zone. It's this area between the 1949 armistice line, which runs like this, and the wall, which established new unilateral borders. This is the seam zone here. So these areas are contested, and that's actually one of the reasons why they're still green and full of mushrooms instead of like people. I mean, there's evidence of people everywhere. The history is just in the landscape. And so the mushrooms are growing around these old agricultural buildings or amidst tear gas canisters. Some of the areas in the valleys are actively managed or even fought over. Like, while I was there was a group of settlers that set up a new outpost. But many of the olive groves haven't been tended for at least a generation, and so pines and oaks are growing up through the terracing, which is hundreds of years old, at least, if not thousands. And the reason I'm showing you all this, rather than just diving directly into the biology and talking about like karyogamy or polyploidy or whatever, is that for mushrooms, context just really, really matters. Like, the only part of the organism that we ever see, the mushroom, is the reproductive organ, and the rest of the thing lives underground and forms these really specific ecological relationships with trees or other fungi. And the mushrooms that we can see and harvest and try to identify, they're often extremely similar to one another. They're like tiny and brown, and the only way to differentiate them is to pay really close attention to the landscape that you found them in. What kind of trees were they associated with? What kind of soil? Like this one, Cryptomerasimus cobariensis, it grows exclusively on fallen olive leaves. These are the olive leaves over here. The mushrooms fruit when it rains, and the entire life cycle lasts just a few days, and only in these very specific places. Or this bolete here, which grows in association with oak. You can see the oak leaves at the bottom. Boletes are a really large group of fungi, which usually have this sort of spongy spore tissue instead of gills. Here's another one in cross-section. And boletes are really cool because they form what's called ectomycorrhizal relationships with other plants. So you can see the hyphae network of this fungus forming a sort of sheath around the root of this plant. Here it is more zoomed out. And what happens is that the hyphae in this sheath end up penetrating deep inside the root and come to occupy the intercellular space between the root cells surrounding them. This is a symbiotic relationship, so when the bodies of the two organisms occupy the same space like this, they can exchange minerals and sugars and communicate. So, before we move on, there's one more section of wall that I want to show you. This is in the middle of Bethlehem, walking distance from where I went to work every day. You can see in this corner how there's a couple of parts of wall that have come together to create a kind of like narrow passageway. It's a one-lane road, accessible only on the Israeli side, and it penetrates quite deeply into Palestinian Bethlehem, creating this kind of like part fortress, part pilgrimage site around Rachel's tomb. This is Rachel's tomb right here at the top of this old Palestinian cemetery. They left the cemetery on the Palestinian side of the wall, just taking the tomb, and soldiers patrol along the rooftop. This is the tomb right here. It's actually kind of hard to see the building today because it's been so fortified, but here's what Wikipedia says it looked like in 1894. The shrine is much older than that. It's supposed to be the spot where Rachel, wife of Jacob, was buried. This is what it looked like in 1933. And here are some soldiers at the tomb, sometime after the West Bank was occupied in 1967. And here are those same pillars today. They've been totally enclosed by the fortifications. So when you drive in, you have to go through this sort of like narrow passageway with the walls arching up above you, and then you're led into this parking lot complex, also surrounded by walls. Everything else has been cleared away. Of course, this wasn't always a parking lot. There used to be buildings here. When the IDF was occupying this area, they designed the wall to enclose certain dwellings. So these houses here are occupied by Orthodox Jewish families who, for reasons that I really wish I could empathize with better, have chosen to raise their kids in this place surrounded by walls on all sides. The tomb itself was originally built on this major thoroughfare, Jerusalem-Hebron Road, which runs like this. And so when they built the walls, all the traffic had to be rerouted. You can see all these cars making U-turns here. Here's what that looks like from the ground. But, of course, the settler road to Jerusalem had already been built, so the IDF was unconcerned about blocking the original road. They just repurposed the roundabout to guide traffic inside the compound. But for Palestinians who have to interact with the wall every day, well, it's just kind of everywhere. If you want to reach the cemetery where the tomb was, you can walk down this path following the perimeter of the wall. There's one corner of Rachel's tomb on the left here. I would pass by that spot every day, and it would always give me this, like, scared, claustrophobic feeling, like the sky was going to disappear. But there are some people on both sides of the wall who choose to live their lives surrounded by it. Like, they could leave, move, and go somewhere else. There's a lot of people who would be quite pleased if they did. But they don't. They stay where they are. And I tried to understand why. I mentioned that there was an Orthodox Jewish family living inside the wall here. There's also a Christian family that lives here, outside the wall, but just as surrounded. I spoke with a woman who lives in that house. Her name is Claire Nastis, and she told me this remarkable story of staying in her home even as the IDF was... they tried to set up a military base on her roof, and these Palestinian homes, they've got these like big iron doors that lead up there, and the door was locked and no one in her house had the key, so the IDF, they just decided to bomb it, because that's what you do when you're a military. And Claire, well, I'll let her tell you.
The soldiers who were staying and camping in a camp behind my building, the commanders were two with other soldiers. Usually they have one commander, but this time they came two. One tough lady, they usually be tough to be commanders, and one man. So they decided to set up a military in my bedroom, in my children's bedroom, and on the roof. And they didn't accept to wait. So they decided both to bomb that iron door on the roof, why they caged our 9 children on the stairs, steering their guns and cocked it toward their heads, and one soldier toward my head and other sister-in-law head. We were standing there watching, and this is the place where they caged us and left the old sick mother inside the house, inside my apartment up there. And the roof is very close to the stairs, to where we were caged on the stairs in front of my apartment. It's one stair up. The commander man stood on the stairs watching us, and he sat there waiting for the commander, tough lady, who left with other soldiers to bring the bombs from the camp. And it took her about 20 to half an hour minutes, 30 minutes to come back. During this time, I was really praying, asking the Lord to speak through my tongue. I do that often, all the time, just to save my children, our family, because they were crying and yelling from having guns toward their heads. As children from 2 years old till 12 ages, it was really a terrible thing. I couldn't believe that also we should accept or bear having a iron door bomb in the same building while the children were there. So I start talking to that commander and looking at him. First he denied, we shouldn't. And the soldier cocked the gun toward my face to stop it. But I didn't listen. I never afraid, because this is what God said to us. If you believe in me, you should not afraid or have any fear. Because trust me, I will protect you. So I do this all the time. That's what saved our life, really, in Americans. So what happened, that commander, I stopped me, but I continued asking him politely. I told him, I'm just asking politely, why you are denying to talk to me? Just answer my questions. I wanted just to ask if you are really a real religious Jewish believer and believe in God. He was proud to say yes. I told her, oh gosh, how come? You should prove it for yourself first. I told him, do you have children? He couldn't not to answer me. He said, yes, for sure I do have 3 children. I told him, okay, I will take your gun now. He was carrying the gun, of course. And go to your home and point the gun toward your 3 children's heads. What you will do with me? He said, oh, how dare you could say that? I will kill you while just to be close to my building, not to allow you to enter. I told him, but you are inside, but I didn't kill you. And you should think of that and look at your heart. If you accept your children having this case, how should I accept having guns toward our children, 9 children heads, not 3, and you are just simply decided to bomb a door, an iron door, in front of them? What kind of fear you are doing, and how you really belong to God? And I looked at his eyes. I've seen something different and changed. And he looked at his inner peace and heart. I could feel it and see it from the Holy Spirit. And imagine what he did when the lady commander came bringing and carrying heavy bombs, because should be a big bombs to bomb that iron door. You can imagine how thick it is. Yeah. So she was carrying it, asking his help to go just up one stair. I followed them kindly, without showing the soldier that I'm fighting, just to watch what he did. What he did, she was putting the bombs on the door, asking his help. He was taking it off. And he moved all the bombs, and he didn't accept. He changed his mind. She fought him, actually. She really jumped and yelled. She promised to kill who changed his mind, because it shouldn't be as a commander. But with God, we could really protect our family, and he kicked out all the soldiers from the building.
Wow.
And I was thanking him, and I brought the paper that they gave it to us, showed that they have the right to enter anytime, even in midnight we should open it, otherwise they have the right to bomb the door and enter. It's really a terrible life, a horrible fear life. I asked him to let me meet that commander who signed this paper, it was him. And he said, I promise not to come back during my period. And it was another, only 2 months we were resting from during those years of having them in. So yeah, that's what we could do.
أشهد أن لا إله إلا الله أشهد أن لا إله إلا الله أشهد أن محمد رسول الله أشهد أن محمد رسول الله.
So, after Claire told me that story, she gave me this little nativity scene. It's Bethlehem, after all, so there's nativity scenes everywhere. Here are two of the wise men, and it's blocked by a wall. Oh, yeah, I'll show you this in a second. Here you can see there's a little baby Jesus and Mary and Joseph, and yeah, the Roman Empire built this wall to keep baby Jesus away. So because I'm American, I actually had certain kinds of freedoms that neither Israelis or Palestinians had. Like, I could just get in a car and drive through the checkpoint and go through that narrow passageway and visit Rachel's tomb from the Palestinian side. And while I was in there, there was this man, this Orthodox guy, and he looked at me and then he gave me this book. It's called Holy Nation. Holy Nation, a guide to Shmiras Habris. Shmiras Habris is Hebrew for not masturbating ever. Yeah. And inside there's this section called Make Fences, which reads, by making boundaries, one is guaranteed to stay far away from temptation. Where there are fences around sexual immorality, there is sanctity. So mushroom sex is really weird because for most of their lives, fungi exist in what mycologists call a multinucleated polyploid state. That's jargon for saying that inside the cells of the fungus there are usually two, although sometimes there are more. Fungi have multiple nuclei, and these nuclei are separated by their own nuclear walls even though they occupy the same cell. It's quite weird. And the nuclei, they operate largely independently, and they come from separate sources. So what you can see in this diagram here is that this fungus was formed when these two hyphae networks, which started over here, fused together, and now the only cells that the network can grow are occupied by these two different nuclei. You can see here's the black one here and the white one there. So when it comes time for that fungus to reproduce, to have sex, it won't actually look for an external mate outside of itself. Instead, the fusion will come from within its cells in a process called karyogamy. The first thing that happens is that those segregated nuclei that have spent the entire life of the fungus independent from one another, they need to fuse. So their nuclear walls dissolve, and what's inside begins to mingle. And then the chromosomes from each nucleus align, and then pair with a partner from the other nucleus. And what happens next is this really beautiful process called crossing over. What happens is that parts of the chromosomes, they break off, and then they reattach themselves to the other chromosome that they've been partnered with. And the effect of this is that the genetic identity of the cell is rewritten, basically. It's a chimera now, and all of that DNA that was kept separate and organized in those two different nuclei, it's now totally mixed together. And then it gets duplicated and repackaged into these teeny tiny spores. That's what a mushroom is, actually. It's a sexual spore-producing organ. And the spores are made in the gills, and then they come out and they spread through the air all over Palestine. Here's a picture of some spores I gathered under a microscope. They're really cute, and I love them all. Hold on, just 1 second. There's something really cool I want to show you. So these are button mushrooms, Agaricus bisporus. They're the kind that you can get at the grocery store. And I left them out on this paper overnight so that I could show you some spore prints. And I haven't looked at them yet. I really hope they turned out well. Let's take a look. Wow. Oh, wow. They're just like these. So you can see the structures of the gills here, all these little lines. And the spores come out of the gills right there, and then they map right on, and there's just so many spores. Like, look how dense this is. Look at all those spores on my fingers, around my fingerprints. I wonder if it will make good eyeshadow. Can you see that? Is that doing anything? Maybe a little bit. These mushrooms, they make thousands, millions of spores. And the spores, they just come out of the gills and flow through the air, and then they land where they land. It doesn't matter on what side of the wall they're on or what a border is. Like, if you were a mushroom, would you care about borders? Mushrooms, they just adapt themselves. They mold themselves to whatever kind of landscape they're in. They're molds. Like, when I was in the desert at this monastery called Marsaba, I found this tiny little clitosobe on the desert cliffs. It was so tiny, and its gills were really thick to avoid water loss. Or here, in the village of Batir, this is one of the few remaining places in the West Bank where traditional terrace agriculture is still practiced, and the soil there is crazy fertile, and I found very many Lepista sordida among the olive groves. This was literally the only place I surveyed where I could find edible wild mushrooms in large enough quantities that I could make a meal, and they were so tasty. So I started this video by showing you this paper, right? And I said that this video was going to be about how I wrote the paper so that if you wanted to, you could write a paper like it too. And that's still the case, but so far this video hasn't really been about papers, right? It's been about context. The thing about these peer-reviewed papers is that they come from institutions whose job primarily is to give this kind of like aura of credibility or of scientific objectivity to themselves, and by proxy to the authors that write them. That would be me looking at an objective scientist right here. There's my name in black and white. But the thing is, I've never really felt like a scientist, and so it's been quite a challenge for me to introduce my work to you in this way by outlining my materials and methods and then reporting the results. In a lot of ways, identifying mushrooms is pretty simple. These organisms have names, and if we pay careful enough attention, we can figure out what those names are in a way that basically everyone will agree with us. And that's what science is, in my opinion at least. It's creating knowledge in such a way that it will look the same from anyone's perspective. But the thing about fungi is that they're not the kinds of creatures that will look the same from all perspectives. They're extremely variable, and because hyphae networks are just a single cell thick, they're massively affected by their environment. Every cell is always already surrounded by other living things all the time, other hyphae or plant roots or microbes. And I didn't have access to any genetic equipment when I was doing this research, so I found myself constantly needing to refer to these other forms of life that were around me if I was to have any kind of understanding about what the mushrooms were at all. And so, in order to do my job right, I just had to pay really close attention to, well, context and to these other living creatures that surrounded me. And the living things that were around me the most, actually, were humans and this terrifying wall. And that affects the mushrooms, right? Like, objective research paper or none, I was just caught up and surrounded by this geopolitical conflict.
Leve Palestina och krossa sionismen. Leve Palestina och krossa sionismen.
So they're gathering up there.
Leve, leve, leve Palestina. Leve, leve, leve Palestina.
The protest is about to begin. Behind me, there's maybe 100 people gathering at the checkpoint.
Leve Palestina och krossa sionismen. Leve Palestina och krossa sionismen. Och vi har odlat jorden. Och vi har odlat jorden. 2, 3, 4! 1, 2, 3, 4! Occupation no more! Occupation no more! Vi har plockat citronerna. Och vi har plockat citronerna. Och pressat oliverna. Och pressat oliverna. Och hela världen känner till vår jord. Och hela världen känner till vår jord. Leve, leve, leve Palestina. Leve, leve, leve Palestina.
So, after that, now I am ready to show you how I wrote this paper and how I got it published. I do think that the peer review process is an important one. It keeps the knowledge that we make at a level of rigor and objectivity, and I want there to be more people in the world who can write stuff like this. But as we go through it, please keep in mind that underneath the surface of this paper, with all these very lovely mushroom photos, there's a tangle of experiences and thoughts that didn't make it into the paper and that don't necessarily look terribly scientific or unbiased or even correct, but that nonetheless had a role to play in this process. Palestinians protesting a wall shouldn't have an effect on what mushrooms grow there, but they do, and this paper has always really been about that. So let's go on one more mushroom hunt in the valley before we move on to some of the more technical stuff.
I'm in Amahur Valley, and there is a truly giant agaricus by my feet. I'm gonna pick it up. Look at this thing. It's like buried underneath the soil, and it's just rupturing out of the ground. Look at that. This is quite a mushroom. It's incredible. So here are the 2 agaricus mushrooms, and to preserve them for the museum, what I want to do is take a cross section like this, like maybe 1 inch thick, and then dry it in the toaster oven, so that you get you preserve the whole thing. We're gonna cut a cross section of it so that we can preserve it for the museum. We're gonna dry the cross section. Alright, there we have it. So, normally I would dehydrate mushrooms in a food dehydrator, but we don't have one, so making do with toaster oven. We'll be in here for a couple of hours, and that should be enough to preserve them so that they can be in the museum for a long time.
Okay, so first of all, that wasn't an agaricus. It was a species called Amanita ovoida, the egg Amanita. And I'll get to how I figured out my mistake in a bit, but a big part was taking good samples and preserving them. So whenever I found a mushroom that I wanted to take back to the museum, I would make a spore print and then I would dehydrate it for preservation in the archives. The Palestine Museum of Natural History is a small and mostly volunteer-run organization. Like, I'm the only mycologist that works with them, and so we didn't really have any proper mushroom equipment. So to dry the samples, what I usually did was just put them out in the hallway to dry in the sunshine. You can see there's the hallway over there and some of the mushrooms I was drying. Collected mushrooms were dried slowly over a period of days using solar radiation. Ooh, I sound like a scientist. But that Amanita ovoida was too big to dehydrate with solar radiation. There are these flies that will lay their eggs in the bigger, fleshier mushrooms, and if you don't treat them any further, in a few days your samples will just be these like slimy piles of maggots. It's great. So I didn't want that to happen, so I added some extra dehydrator energy in a toaster oven, and that really made me feel like a scientist. Once I'd preserved the samples, the next step was to identify them, and I did that using Facebook. I've heard that Facebook is a very reliable source of information. So there were a bunch of groups that I would visit to help me identify the mushrooms that I found, and this one was called Mushroom Talk, and you can see here that I wrote, "Could this be Agaricus bisporus, found in a lawn amid grass and fennel and asters?" Whenever I saw mushroom species in the field I didn't know, I just thought it was Agaricus. What a fool! So then the very nice mushroom people in the group corrected me. Dimitar wrote, "Take a careful study of the overall features of the genera. Avoid wild guessing. This can be done very easily if approached systematically. What are the basic key features of genus Agaricus? These are related genera, but still Agaricus has some solid features that sets it apart." And then I replied, "So these don't have an annulus on the stalk. Instead, there's a vulva at the base, so not Agaricus, but the spore and cap coloration makes me pretty skeptical of Volvopluteus." But Dimitar insists this is 110% Volvopluteus. Everything points to it. So then I looked up a bunch of pictures of Volvopluteus gloeocephalus, the species that the mushroom people said it was, and what I found was that most of the pictures available were from North America, and that the coloration of both the cap and the spores was distinctly lighter than what I had found in Palestine. But the rest of the description checked out, like the description of the gills or the presence of the saciform vulva, and coloration is a trait that will vary quite a lot with environment, so I thanked Dimitar and went with his ID. But in the paper, I noted the color difference and then wrote that there were likely underlying environmental causes and that genetic analysis of the samples was required to determine how phylogenetically distinct they are from V. gloeocephalus elsewhere in the world. This kind of thing happened pretty frequently, actually. That's because most of the mushroom species that are easiest to identify have cosmopolitan distributions. That means they can be found on multiple continents. But the type specimens, the ones that get photographed and make it into guidebooks like this, they're usually from Europe or North America because that's where all the mycologists are. Nobody studies mushrooms in Palestine except, like, me, for some reason. So there are all these, like, tiny little morphological variations that are unique to the region that you can find if you look carefully enough. Like, for example, North American species of Lentinus arcularius are described as having this hairy fringe along the cap margin, but none of the samples that I found had it. See, it's just not there. Is this difference adaptive? Does it mean that they're a separate species? Well, no one really knows. Something like this might be extremely important, or it might not matter at all, but either way, what it shows is just how contingent the form of a mushroom is on the place that you find it. Like, you can't really make sense of why a mushroom is the way it is without also trying to make sense of why its context is the way it is. So that's basically what I did. I walked through these landscapes looking for mushrooms, but also just looking at the landscapes themselves and trying to understand how they came to be that way. And I talked to people. I talked to people on the internet who knew more about fungi than I did, and I talked to Palestinians who knew more about their land than I ever will. And then I actually had to sit down and write this paper, so let me tell you a little bit about that. So here is a copy of the paper. It has a fairly typical scientific structure. It starts with an introduction, and then there's a materials and methods section. The results section, which comes next, is a bunch of pictures and technical descriptions of all of the fungi that I found. And then at the end, there's a discussion section. I got to include this evolutionary tree of all of the mushroom species that I identified. I really like these tree patterns. That was cool. And then the final section is a literature cited, which is my bibliography, all of the sources that I used to identify and describe the mushrooms. So, yeah, That's a scientific paper, Macrofungi from the Hebron and Jerusalem Hills of Palestine. This paper was published in a journal called Mycotaxon, which is a journal exclusively devoted to the taxonomy and nomenclature of fungi. This is a pretty specific journal. It's not like Science or Nature or any of the big ones. Mycotaxon only publishes papers specifically about, well, mycological taxonomies. It's got a great name. So I chose it because I knew that the scope of my paper matched pretty closely to the scope of the journal, so it would have a pretty good chance of getting published. But actually getting the paper published is quite a lot of work because Mycotaxon has some very specific standards. This is the sample manuscript that I had to conform my writing to, and the formatting was very specific. In the abstract, I had to briefly summarize research results in text formatted in regular or italic ×8. The introduction cites significant background history and documents research observations in 9 points, stressing briefly summarizes basic protocols. In the technical description, authors may separate individual introduces features used using text, either semicolons or italic, emphasize terms, but be consistent. I had to format all of the photographs I'd taken as JPEGs, 300 dpi, 11 centimeters in width, and then insert them into a separate image document, and then in the main text, I would detail a technical description of the mushroom, including its ecology, its context, as well as any other additional information about the species that I might find in some other peer-reviewed journal. Basically, I had to keep track of 11,000 little tiny details. It was so much fun! And then, once I was done, the next step was to get the paper peer-reviewed. I had to find some mushroom experts who could tell me if I was full of shit. They're the ones who really make you feel like a scientist. I had to send each of the reviewers this reviewer document, which they had to fill out in order to confirm that my formatting was correct and that there was scientific support for all my IDs and other tedious things. And then I waited for over a year until all of the peer reviewers got back to me, and one of them told me that I should cut half of my conclusion section, the part that I was most excited about, about how science could be done by anybody and that it didn't need to be hypothesis driven. Yeah, he said that wasn't relevant, so I cut it. Then, after I made all the edits the reviewers suggested and they confirmed them, the manuscript was then sent along to the Mycotaxon editor for more review. The editor explained to me that I needed to correct some very bad word processing practices and that my literature section was bizarrely formatted. So I went through another round of revisions, and then finally, the paper was published. This page shows all of Mycotaxon's papers organized by geographic region, and you can see here that there's one entry under Palestine, and here's my paper. Yay! I'm a scientist now! But there's actually still one more step to this process. It's the step that we've been in for this entire video, actually. Once your paper is published, it's your responsibility as a scientist to communicate about your work to others in a way that they can understand and enjoy, ideally. We're 40 minutes into this video, so hopefully you're enjoying yourself. Otherwise, I really have no idea why you're here. Basically, I gave a bunch of talks about my work. Here's me giving a talk at the Palestine Museum of Natural History, and this shot is of me at Binghamton University. And when I give talks about this, what I try to emphasize is just how simple and accessible this research is. I think it's valuable specifically because anybody could do it. And also super simple. What can you do with a Palestinian volunteer who's just out of high school? How can you have them go out and do this thing?
It is also valuable.
Yeah, I think so.
Because I keep hearing you say low level and not professional and simple, but this is really acquiring really important background information about the space, and is really valuable no matter where you do it. I mean, you have to start by looking at what's there. So I think you should give it more credit.
I so maybe I'm underselling myself. I certainly believe that to be the case. And one thing that I found really refreshing, this is I guess an epistemological point, but I really liked that the majority of these papers were not hypothesis testing papers. Because I've had experiences being pushed to come up with some sort of falsifiable claim that just doesn't feel meaningful, or that I understand enough to actually ask an important question. We can get very results focused because we want to publish that thing, here's this new piece of knowledge that has never before been known. But I think there are other ways of generating knowledge that aren't necessarily falsifiable. And so just being given the chance to go out and see what's out there, and get that intimate sense of a landscape and an ecosystem, I think is enormously valuable, and isn't often the way we think about scientific research.
Throughout this video, I've had this maybe bad habit of repeating that I don't really feel like a scientist. And what I mean by that is that these days it's become harder and harder for me to have much interest in proving specific things. Like, I don't usually think using falsifiable terms anymore. I used to think that way a lot more often, but I'm different now. Like, an earlier version of myself would probably have spent this entire video trying to prove to you that the wall was an actual living organism, like some kind of parasite that consumes creatures from the inside out. And that's certainly what it feels like to me when I'm around the wall, and I've spent a decent amount of time with fungal parasites. Like, I know what they look like and how they behave. There are definitely some commonalities there, big ones. But that's not something that you can prove. That's not objective. It's a different kind of knowing, one that looks more like an analogy than a proof. I think that when you take the time to really immerse yourself in a landscape and develop an awareness of all of the different kinds of life that surround you, certain kinds of patterns just emerge. You start to notice correspondences between some of the things that are really big and other things that are really small. Like, now I can't look at this image and not think about multinucleated polyploid cells and all their contradictions. Cells that spend their entire lives totally wrapped up around or within other forms of life. Sometimes those intimate relationships can be beneficial, and sometimes they can kill. It's usually both. And you or I or these mushrooms or the state of Israel even, we're not really all that different in this regard. We all need to learn to live with others. So that's why my approach is primarily just observation, no hypotheses. I'm not trying to falsify anything or anyone, God forbid. I just want to understand better all of the different ways that there are to be alive, and maybe also communicate about it with you. Like, that's why I made this video, right? I want to learn about mushrooms with you. So if you want to take some mushroom photos with me in Palestine this winter, or anywhere really, I've left my contact information in the description, or you can just leave me a question in the comments. I'll read it and reply. We'll do the thing. It'll be nice. That's why I'm here. So yeah, thank you so much for watching. It means a lot. And yeah, I'll see you in another video, I guess. This one's all wrapped up. Okay, bye.